Welcome to today's, good afternoon, everyone. Welcome to today's seminar in oncology lecture, which is the Marjorie Beth Salmon lecture. And we are delighted to have with us Judith Balmania, MD, PhD from Val d'Hebron. In September 2013, the late Dr. Walter J. Salmon Jr. made a generous gift to Dana-Farber to establish this annual seminar in women's cancers and genetics, named in honor of his late wife, Marjorie Beth Salmon. Marge served on the Dana-Farber Board of Trustees from 1993 until her passing in 2014. Walter was also a longtime faculty member at the Harvard Business School, teaching consumer marketing and retailing for nearly 60 years before he passed in March 2015. Marge and Walter were married for 60 years, and raised three daughters, Liz, Joan, and Stephanie. And Liz Salmon Frank is a well-known patient advocate in the breast program where she runs the advocacy core. Though Walter passed away before the inaugural presentation of this seminar, we are grateful for his generosity and to the Salmon family for all they have done for Dana-Farber over the years, and we are honored to hold this annual lecture in memory of Marge Salmon. So I would like to introduce Dr. Judith Balmania, who is joining us today from Barcelona, although for her it's tonight in Barcelona. So after finishing a 2.5-year clinical research fellowship here at Dana-Farber with Sapna Singhal and me in cancer genetics, Dr. Balmania returned to Barcelona to the medical oncology department at Val d'Hebron Hospital, to establish the familial cancer program. And since then, she has continued to lead the hereditary cancer genetics group and also to serve as attending physician in the breast cancer unit. She obtained her PhD from the Universitat Autònoma de Barcelona. I'm sorry to say that with such a terrible accent, for her research on a prediction model for hereditary colon cancer. She has had initial and long interest in the genetic epidemiology of hereditary breast cancer and has linked in part her research with Sarah Gutierrez and Orlan Diaz from the Oncogenetics Lab at VHO and HVH. These studies have shed light on the prevalence and characterization of new pathogenic variants and variants of uncertain significance in the BRCA genes and beyond. Dr. Balmania and her team are highly interested in unraveling the role of intronic and splice variants in major genes associated with breast cancer. Once PARP inhibitors were discovered, she became interested in their clinical development as a targeted therapy for patients with BRCA1 and 2 mutations. She has been actively involved in a number of trials and trial steering committees. We have done trials together and been on trial steering committees together. And she has conducted several phase 1 to 3 clinical trials with PARP inhibitors or other DNA damaging agents targeting tumors in BRCA deficiency. In 2012, she began to study mechanisms of resistance to these targeted therapies in breast cancer patients in collaboration with Violeta Serra and Cristina Cruz from the Experimental Cancer Group at Val d'Hebron. And they have been awarded several competitive national and international grants to investigate this field. In collaboration with Violeta Serra, Dr. Balmania has gathered a collection of patient-derived xenografts from patients with BRCA mutations, and on the basis of this work, conducts translational research in mechanisms of resistance to PARP inhibitors and new therapeutic combinations. She has work to discuss on RAD51. And today, Dr. Balmania will speak with us about advances in personalized medicine with the use of PARP inhibitors and novel biomarkers of homologous recombination repair deficiency. Dr. Balmania, welcome to Boston. I wish you were here in person. Oh, before she starts, just if you have questions, please enter them into the Q&A function, not the chat function, and we will have an opportunity for questions and answers at the end of Dr. Balmania's talk. Thank you, Judith.
Thank you, Judy. I'm trying to share my screen. Hopefully it works.
You may need to do it again.
Okay.
No, it's okay. We just see. There you go. Perfect.
Now? Okay.
Yeah.
Excellent. Great. So thank you so much. And good afternoon, everybody. Thank you for this kind invitation and also the opportunity to participate in this seminar. Today, I will be presenting my perspective on advances in precision medicine with the use of PARP inhibitors and a novel biomarker of homologous recombination deficiency. These are my disclosures, including the fact that I'm a clinician and it is 10 p.m. in Barcelona. So the focus of my talk will be to share a personal and historical perspective on targeted therapy for patients with a germline BRCA mutation, followed by the development of a functional biomarker of homologous recombination deficiency for PARP inhibitors, the RAD51 assay. So 20 years ago, as Judy already introduced, I was granted for a clinical research fellowship in the high-risk clinics at Dana-Farber. Judy Garber has been one of my mentors since then. In 2005, I returned to Barcelona, and that same year, 2 seminal papers showing the in vitro synthetic lethality of PARP inhibitors in BRCA deficient cells were published. With the prior evidence that patients with BRCA mutations were specifically sensitive to platinum-based chemotherapy, and actually Judy Garber led some of this clinical evidence, and that potential synergy with PARP inhibitors could happen, sooner than later, we started to think on a clinical trial to develop this combination in patients with advanced solid tumors. So this first patient was enrolled in November 2008, and the publication in Annals of Oncology was achieved in 2014. So the study design included a continuous dosing of olaparib at increasing doses in combination with the maximum dose of cisplatin, 75 milligrams per square meter. As this was not tolerable, we had to shift to an intermittent dosing and reach a tolerable dose at olaparib 50 milligrams PID days 1 to 5 and cisplatin 60 milligrams a square meter. Unfortunately, at that time, we were not ready for such a de-escalating regimen, and we abandoned this approach. But I wanted to give you some data from this trial that help us and provide us a lot of learnings and lessons learned. The study population included 54% of patients with a known BRCA1 or 2 mutation, 78% had breast cancer, and they were heavily pretreated, with a median 3 prior lines of chemotherapy and a range between 1 and 11. As you can see here in the waterfall, the anti-tumor efficacy was notable, with 41% of patients achieving a confirmed response. But more interesting, the overall response rate in BRCA breast cancer patients was 71%. And I think it is also worth to highlight that olaparib was administered as maintenance in this phase 1 clinical trial after a maximum of 6 cycles of cisplatin, or if the patient had to interrupt platinum due to toxicity. And 13% of the overall cohort had durable responses for more than 1 year. In 2011, this was really quite astonishing. I believe we were the first ones to provide maintenance with platinum therapy. I would like to give you a real example of a patient who was enrolled in this trial. She was a lady with a BRCA2 germline mutation who had had bilateral triple negative breast cancer with adjuvant chemotherapy and 3 prior chemotherapies in the advanced setting. She received the combination for 6 cycles and then followed by olaparib. This is her baseline picture of the cutaneous and skin involvement, and she achieved a partial response with an 85% reduction, and the duration of that response was almost 3 years, which is really an amazing long period of time. In fact, another patient with a BRCA mutation and a triple negative breast cancer is studied in 2009, and she's still on treatment with olaparib 200 milligrams BID, 12 years later. So she's the longest responder I've ever treated. For some of you who are probably very young, you don't know how challenging and difficult has been to keep with the development of PARP inhibitors in breast cancer. Actually, around 2010, 2012, the clinical development of PARP inhibitors, especially in breast cancer, was halted. There were multiple challenges. I'm sure Judy remembers them as well, in different settings, and that had to be overcome before proceeding with the clinical development of targeted therapies. Germline testing itself was one of these challenges. In 2011, we were invited to write a mini review to report these stumbling blocks on PARP inhibitor development for BRCA1 and 2 associated breast cancer. And the field, thanks also to the efficacy observed in ovarian cancer, moved forward. Nevertheless, in the meantime, in 2011 approximately, PharmaMar, which is a pharmaceutical company from Spain working with molecules obtained in the sea, was developing lurbinectedin. Lurbinectedin is a trabectedin analog that inhibits active transcription by RNA polymerase II degradation, and it also generates double-strand DNA breaks. It attaches to the minor groove of the DNA of the helix, and it causes a stalling of the replication and fork and transcription. Therefore, a deficient homologous recombination repair pathway would make these tumors more vulnerable to this agent. And of course, patients with a germline BRCA1 or 2 mutations would be exquisite candidates. So a phase 2 trial was studied in BRCA1 and 2 associated or unselected metastatic breast cancer population. Centers from Boston as well, several centers from Spain started to enroll patients in this phase 2 clinical trial. And as you can see here, the overall response rate was 41%, again in a heavily pretreated population, and reaching 61% in BRCA2 mutation carriers, and also observing benefit regardless of hormone receptor status, and interestingly, with a better efficacy in patients with no prior platinum compared to patients with prior exposure to platinum. Also, during this phase 2 trial, there was a change in the dose, changing from a flat dose of 7 milligrams, a flat dose to 3.5 milligrams per square meter, and that improves safety. But as you can see here in this forest plot of predictors of response, PARP inhibitors, the exposure to prior PARP inhibitors was one of the main negative predictive factors of response to lurbinectedin. And actually by then, around 2016, approximately, we started to have data of the efficacy of olaparib and talazoparib, and actually Olympiad and Embraca were then published in 2017 and 2018. So it was kind of challenging for this drug to identify in which position was going to be placed with no responses after PARP inhibitor. In addition, despite this nice overall response rate, the median progression-free survival didn't overcome 4 months. So we were seeing that responses were good, but progression-free survival was quite short. So here is where we started our interest in studying mechanisms of resistance and predictive biomarkers to targeted therapies against BRCA1 and 2 mutation carriers. We then teamed with one of the translational research groups at VIO, the experimental therapeutics group led by Violeta Serra, to work in this field. So at that time, we were mainly working with patient-derived xenotransplants, which were one of the most common in vivo preclinical models that were available at VIO. And this allowed us to study biomarkers and also to correlate results between the mouse avatars and patients. So in 2011, we started a collection of BRCA PDX models at VHIO under the leadership of Violeta Serra, and then together with Cristina Cruz, Marta, and Alba. We also established a research process to obtain pair samples of metastases before treatment and at progression of patients undergoing targeted therapy and a BRCA mutation. So at this time, we were enrolling patients in the lurbinectedin trial, and we started to obtain these paired biopsies. So knowing that the NERS pathway had been already described as a mechanism of resistance to trabectedin, we focus our analysis on doing whole exome sequencing and looking for mutations in this group of genes as well as other homologous recombination repair genes. And we did this in pre and post-progression tumor biopsies of patients treated with lurbinectedin. Here, I'm showing two examples of acquisition of mutations in the ERCC4, a gene encoding for the XPF protein. In the left, here you can see a patient who did not have any germline nor somatic mutation pre-treatment in the ERCC4 gene, but then acquired at progression a somatic mutation in ERCC4 with a variant allele frequency of 47%. This mutation was demonstrated to cause resistance to lurbinectedin in the in vitro cell proliferation assay. In the right, another patient who had a germline alteration in ERCC4, who was enriched to homozygosity in the primary tumor. This patient had actually primary resistance to lurbinectedin, and then the PDX also was completely homozygous for this mutation. Interestingly, none of the patients who were long responders to lurbinectedin never acquired a mutation in ERCC4 or other NER pathway genes. Finally, prior data had also suggested that the NER alterations could induce resistance to lurbinectedin, but increase sensitivity to cisplatin. We actually confirmed this in an in vitro cell experiment in a cell with an ERCC4 mutation. And additionally, also in the clinics, in a patient who acquired two mutations in the NERF pathway, we observed, this is the patient who responded to lurbinectedin and then at the time of progression acquired these two mutations in the NERF pathway, and then she received cisplatin after progression and she obtained again a partial response. So we did have both in vitro and in vivo demonstration that the NERF pathway alteration was being a resistant mechanism to lurbinectedin, but also a predictor of sensitivity to platinum. This collaboration was finally published at JCO in 2018. So now I will change gears to PARP inhibitors. Just a brief summary to remind you all that DNA single-strand breaks are normally repaired by PARP-dependent repairs and resulting in cell survival. If PARP is inhibited with a PARP inhibitor, then this base excision repair system will be blocked and the single-strand breaks will remain unrepaired. And when the cell enters into the synthesis, into the S phase, this will promote double-strand breaks. Additionally, the trapping of PARP by the PARP inhibitor also causes stalling of the replication fork, and it also causes these double-strand breaks. So these double-strand breaks are ideally repaired by homologous recombination repair, which involves BRCA1, BRCA2, and several other proteins to enable a high fidelity DNA repair. But in cases where homologous recombination repair is deficient because of mutations in BRCA1 or 2 or other genes, this fork collapse cannot be repaired and the cell undergoes cell death. So by then, in 2017, 2018, the results of the phase 3 clinical trials, Olympiad and BRCA, were published, and germline BRCA1 and 2 mutations were the only biomarkers currently approved and the most robust ones for treatment with PARP inhibitors in breast cancer. The overall response rate was around 60%, and progression-free survival was 7 and 8 months versus 4 and 5. Nevertheless, it must be noted that this molecular selected population of BRCA germline mutations accounts only for 2.5% of the overall burden of breast cancer. And also platinum-resistant tumors were excluded from these trials. So therefore, there was a clear need, and there is a clear need to expand the proportion of patients who may benefit from PARP inhibitors beyond germline BRCA mutation carriers, and also to improve this clinical efficacy, maybe by better selecting the patients who may benefit the most of a treatment with a PARP inhibitor. So the importance of the biomarker, in fact, is that homologous recombination deficiency is at the end the true biomarker of sensitivity to PARP inhibitors. Therefore, knowing the prevalence of homologous recombination deficient breast cancer is key to gain insight of the potential use of this targeted therapy. In this regard, according to the genomic signature HR Detect in the paper by Davis, up to 8% of cancers without a BRCA mutation are homologous recombination deficient. In primary triple negative breast cancer in the GEPAR-6 trial, approximately 42% of tumors without the BRCA mutation are homologous recombination deficient, according to my choice. Based on the knowledge that RAD51 protein is a protein essential in the final part of homologous recombination repair through the homology search and stranded change, we improved a prior existing assay to look at functional homologous recombination repair by the formation of RAD51 foci in the cell cycle phase of DNA synthesis, when homologous recombination repair takes place, because at that time, the sister chromatin is available as a template. So RAD51 foci formation had been proposed already by as a functional biomarker of homologous recombination repair deficiency and treatment response, but previous reports had suggested that there was the need to cause an induction of DNA damage in form of irradiation of the biopsy or administration of chemotherapy in order to cause DNA damage and observe the RAD51 foci formation. Here you can see the correlation between low RAD51 foci and pathological complete response to a standard chemotherapy. So what we did was to develop our immunofluorescence assay of RAD51 without the need of causing an extra DNA damage, but selecting the cells that were proliferating, therefore geminin positive, and that carry enough DNA damage, intrinsic DNA damage, by analyzing gamma H2AH. And this assay is performed in archival formalin-fixed paraffin-embedded tumors to detect gamma H2AH, BRCA1, and RAD51 foci in the SG2 cell cycle phase. RAD51 is a score, is quantified according and counting the percentage of cells in SG2 phase harboring at least 5 nuclear RAD51 foci. A RAD51 high score would mean that there are at least 10% of cells with 5 or more nuclear foci, and a RAD51 low score would include less than 10% of cells with 5 nuclear foci. Here, you have a very clear image by immunofluorescence of a section scored as RAD51 high, therefore homologous recombination proficient, and here a section scored as RAD51 low, and therefore homologous recombination deficient. So what biomarkers of homologous recombination deficiency do we have? So there is the option of sequencing homologous recombination repair gene alterations in the tumor, but these require a biallelic inactivation of an alteration that causes lack of function in a gene that is actually relevant for homologous recombination repair. There is also the option of analyzing genomic scars based on SNP genotyping, next generation sequencing, or whole genome sequencing, and looking for different tattoos derived from genomic instability caused by an error-prone repair mechanisms. And there is the option of performing a functional test, such as a scoring of RAD51 foci in the nucleus of proliferating cells. I will just provide a brief summary of genomic scars and signatures that they have, since they have evolved as a composite biomarker of genomic alterations that specifically identify which tumors are deficient in DNA repair by homologous recombination. For instance, the genomic instability scored by immediate takes into account the proportion of LOH along the genome, the non-translational allelic imbalance, and the larger scale transitions. HR detect is another genomic scar developed by the group of Serena and Nick Sainal, and is composed of 5 mutational signatures, including base substitutions, indels, and rearrangements. And finally, the Foundation Genomic LOH computes the percent of LOH in the tumor genome using the genome-wide copy number profile and the minor allele frequencies of the SNPs. All 3 genomic signatures include the tumoral BRCA1 or 2 mutation as part of the definition of homologous recombination deficiency. So as a summary, when talking about the different homologous recombination deficient assays, the etiology of homologous recombination deficiency can be investigated by means of analyzing the mutation or the methylation sequencing, for instance, methylation of BRCA1 promoter. The current homologous recombination status can be analyzed by using functional tests such as RAD51 foci, and the prior homologous recombination deficiency exposure by looking at the genomic scars. So actually, a key difference between these 2 last biomarkers is that RAD51 is actually a dynamic biomarker, is dynamically and functional, and the genomic scars are in fact an static biomarker. So I will now focus on the preclinical validation of the RAD51 assay in our cohort of PDXs, more than 100. And among them, 42% are homologous recombination proficient and 58% are homologous recombination deficient. Among these, the majority are related to a BRCA1 alteration or a low expression of BRCA1. 13% are related to a mutation in BRCA2, and then 3% related with mutations in PALB2, and then another 2% related with other homologous recombination repair altered genes. So in this cohort of PDXs, what we have done is investigated the underlying mechanisms of resistance to PARP inhibitors. And in the group of BRCA1 deficient PDXs, we've seen that 60% of them are resistant to PARP inhibitors. As you can see here, shown in the pie chart, approximately 1/3 of these BRCA1 deficient resistant PDXs, the cause is related to a BRCA1 hypomorph protein or expression of BRCA1 foci, or the combination of BRCA1 hypomorph with 53BP1 pathway dysregulation. Another third remains unknown and actually related with low expression of BRCA1. And the rest are explained by 53BP1 dysregulation and non-BRCA1 mechanisms, or finally, this still pending investigation of mechanisms that are RAD51 independent. So one of the most interesting and encouraging results that we had when testing PARP inhibitors in this cohort was to observe the good correlation between lack of RAD51 foci and sensitivity to PARP inhibitors, as you can see here in this waterfall in the left. So you have here in gray those PDXs that had a low score of RAD51 and the correlation with response to PARP inhibitor. On the other hand, we were able to do the analysis of different genomic scars, such as my choice or HR detect in some of these PDXs, and we observed that this correlation was less strong between genomic scars and PARP inhibitor sensitivity. You can see also this correlation here on the right, where tumors with BRCA1, BRCA2, PALB2, or low expression of BRCA1 have the most of them low RAD51 foci and are sensitive to PARP inhibitors. So we performed a comparison of accuracy to predict PARP inhibitor sensitivity among the different biomarkers in this cohort of PDXs, and conclude that the specificity decreases with analysis of gene mutations and also with genomic HRD score compared to RAD51. And this finally impacts on a lower accuracy for these two biomarkers compared to RAD51 to predict sensitivity to PARP inhibitors. So now moving into clinical samples, here you can see that RAD51, we have been testing this foci in different tumor types in more than 500 human breast cancer samples, as well as in ovarian, endometrial, and prostate cancer samples. And interestingly, what we have seen is that among the breast cancer samples, as you can see here, the range of the prevalence of homologous recombination deficiency according to RAD51 ranges from 100% in primary germline PALB2 mutation samples to 94% in germline BRCA1 or 2 mutation samples, then 77% in breast cancer patients with a family history or young onset diagnosis, and then 54% among primary tumors with a triple negative breast cancer phenotype. And this likely correlates what we are seeing now also in the clinic. And here, as an example of a functional dynamic biomarker, we can observe in this series of germline BRCA1 and 2 mutated breast cancer that the prevalence of homologous recombination deficiency is 92% at the time of diagnosis, 86% in the surgical samples. Some of them have received prior neoadjuvant chemotherapy, and 63% in metastatic samples. So this is providing us with a clear view that homologous recombination deficiency may evolve as a consequence of therapeutic pressure and also cancer evolution. And that the best measure for therapeutic decision making will be the one will be by using the biomarker performed in the nearest sample before treatment, since the tumor might be evolving. So now I will present the results of 3 clinical validations of the RAD51 test performed in ovarian cancer, in prostate cancer, and in a cohort of triple-negative breast cancer. So the validation of RAD51 in the Chiva trial was led by Dr. Alexandra Leari from Gustave Roussy in Paris. And it was done in ovarian cancer samples who were treated with platinum in the neoadjuvant setting. In this cohort, RAD51 was evaluable in 90% of the patients, and 55% were homologous recombination deficient by RAD51. Tumors with RAD51 low or negative had a higher response rate compared to platinum compared to tumors with RAD51 positive, 68 versus 37%. And in fact, this response rate also translated into a longer progression-free survival, 21 months versus 14 months. And more specifically, among BRCA mutated patients, 67% were RAD51 low, and they responded better to platinum, 75% versus 17% among those germline BRCA mutated ovarian cancer patients with RAD51 high. So in conclusion, here we see that RAD51 could stratify patients for platinum sensitivity, even among the BRCA population. In patients with castration-resistant metastatic prostate cancer, in the PARP-B trial that was testing olaparib versus a standard hormone therapy, we observe here that the RAD51 foci was low in all BRCA1 or 2 mutated samples, only in 1 sample of an ATM mutation carrier, in half of the CDK12 mutated samples, in 3 out of 4 of the PALB2 samples, and in only 1 sample from a patient with other homologous recombination repair gene alterations. So here we can also see in this validation in PARP-B that RAD51 score also differentiated median the median progression-free survival among patients with RAD51 low versus RAD51 high who had a worse prognosis. And also it correlated with overall survival, with a better survival among patients with RAD51 low. Finally, in the GEPAR-6-2 trial, this is a trial that enrolled patients with triple-negative breast cancer to neoadjuvant treatment with or without platinum. And here, RAD51 was scored in TMAs. And it was successfully scored in 67% of this TMA course. And 61% of them had low RAD51 foci. You can see here on the waterfall that also low RAD51 foci correlated very nicely with low expression of BRCA1 protein. in Gepard-Sixto, so good concordance between tumor BRCA status and RAD51 foci was observed. The concordance rate was 65%, as well as with the genomic score HRD, the HRD score by myriad with 87% concordance. 93% of the mutated samples had low RAD51 score. But interestingly, 45% of these non-mutated triple-negative breast cancer patients also had low RAD51 score. Therefore, this might be broadening the identification of cases potentially sensitive to platinum or to targeted therapies to homologous recombination deficiency beyond BRCA1 and 2 germline status. We hypothesized that discordant cases might be the consequence of the absence of BRCA LOH, making that some of these tumors might have recovered homologous recombination functionality despite being mutated. And then these other cases might be related to mutations in other homologous recombination repair genes or epigenetic alterations. On the other hand, 86% of tumors with a positive genomic HRD score also had a low RAD51 score, and 14% were RAD51 high. Here, we hypothesized that discordant cases might be related to homologous recombination repair restoration, or maybe genomic instability of non-homologous recombination repair origin, or maybe a tumor heterogeneity that is not being captured by the genomic scar. So when investigating the predictive value for response to neoadjuvant platinum, we observed that RAD51 differentiated those tumors more likely to respond to carboplatin, 66%, versus 31% here among those with a high RAD51. In the multivariate analysis, the genomic HRD score did not statistically predict the benefit of carboplatin, but the RAD51 low score had an odds ratio of 7. However, these results that were published in Annals of Oncology very recently did not translate into any difference in long survival outcomes related with platinum treatment. We also had the opportunity to work with Nick Turner in the Rio phase 2 window trial, testing the clinical activity of rucaparib in 51 primary triple-negative breast cancers, 8 of them with a BRCA1 mutation. And rucaparib was administered for 12 to 14 days with paired biopsies and circulating tumor DNA sampling. Different biomarkers were studied to predict response to rucaparib. And here you can see that RAD51 test identified BRCA1 and BRCA2 mutation carriers, also the PALB2, a patient with two patients with BRCA1 promoter methylation. And also there was a very good correlation between homologous recombination detect, between HR detect and the RAD51 foci. In terms of response, RAD51 low tumors had a greater circulating tumor DNA suppression on rucaparib after 14 days than RAD51 high tumors. So this correlation was very nicely observed in this very small sample series. And our final validation analysis so far, it is in the PetraMAC trial led by our colleagues from Norway. And in this trial, olaparib was administered for a maximum of 10 weeks in the neoadjuvant setting in triple-negative breast cancer patients before starting chemotherapy. RAD51 score predicted the clinical response measured by MRI. As you can see, patients with low RAD51 were the ones who achieved more response measured by MRI compared to patients with a RAD51 high score. And also on the right, you can also observe here in more detail patient samples with RAD51 foci low, and they are correlated with complete response or partial responses, all but one that progressed despite the RAD51 low foci. And this correlation was better than the one tested by mutations in homologous recombination genes, BRCA1 methylation, or BRCA signature. Here you can observe an image of a patient who was olaparib responder with RAD51 low foci, also BRCA1 low, and an important DNA damage with foci of gametes2AH very high, and then non-responder patient to olaparib with RAD51 foci and also BRCA1 high and gametes2AH very high. So in summary, genomic scars can very well measure homologous recombination repair deficiency in patients with germline BRCA1, 2, or PALB2 mutations, also with patients with other somatic mutations. But the major limitation of these DNA-based assays is the lack of specificity and the low positive predictive value in tumors that have restored homologous recombination repair as a mechanism of drug resistance, since genomic scars are like tattoos and therefore they are not erased from the genome. So RAD51 foci would in fact be a dynamic biomarker and genomic scars a static biomarker. So in conclusion of my talk, I would say that PARP inhibitor sensitivity is mainly dependent on homologous recombination deficiency, and for which different tests are available. RAD51 is a functional homologous recombination deficient biomarker. It is visible in tumor samples from ovarian cancer, prostate cancer, and breast cancer. Is highly concordant with tumor BRCA mutations in early triple-negative breast cancer and prostate cancer, and it's also highly concordant with myriad my choice in early triple-negative breast cancer. RAD51 foci predicts platinum response in ovarian cancer and triple-negative breast cancer, and it is associated with higher progression-free survival in castration-resistant prostate cancer, and also with a higher response in early triple-negative breast cancer treated with olaparib. It is likely that in metastatic tumors, these functional homologous recombination deficient tests are needed, and maybe also in the neoadjuvant setting if we want to be doing a very good selection of patients who might be candidates for de-escalating chemotherapy and receive PARP inhibitors. Just to conclude, which are our next steps? On one hand, we will be starting a phase 2 trial in the metastatic setting, a small trial with 2 cohorts, just to prove that RAD51 might capture patients with a germline or somatic mutation in BRCA1, 2, PALB2, or RAD51 C and D mutations. And in cohort 2, patients will be wild type for any of these genes, and they will be selected based on a RAD51 low score. Patients will be treated with olaparib until progression disease or toxicity, and the primary endpoint, in fact, will be overall response rate. And in cohort 1, RAD51 will be tested retrospectively to analyze its predictive value. We are also working on improving the availability of the RAD51 test through analysis of CTCs and in liquid biopsies. And in fact, this is a big endeavor now ongoing. And finally, we want to continue studying mechanisms of resistance to PARP inhibitors, specifically those that restore homologous recombination repair. So those tumors that become RAD51 high, either by the presence of PRCA1 hypomorphs or the presence of reversion mutations or loss of PRCA1 promoter hypermethylation or DNA damage repair rewiring by the play with 53pp1. And we will then test these new therapies, first in the PDXs or combinations of therapies in patients with RAD51 high tumors. I want to thank you all for your attention. I also want to give my thanks to the group led by Violeta Serra with Alba Job as the postdoctoral and senior scientist at this time. My colleagues in the clinics, Cristina Saura, Joaquin, or Ana Wagnin, Orlan and Sara Gutierrez in the oncogenetics lab, my colleagues in the US, with whom I've learned a lot, and I'm collaborating and also have a very nice friendship with many of you. Mark O'Connor from AstraZeneca, many other co-authors and funding agencies. And thank you to all for your attention, and I'll be very happy to take any question. Thank you.
Thank you, Judith. That was really very interesting and provocative, and it's really a challenge to try and develop a biomarker that you can use to predict reliably response. I mean, here we're so happy that we have something when we have a germline mutation or a somatic mutation, but as you point out, that's certainly not the whole story. So while we wait for people to ask their questions, I'll just ask you, so I was really pleased to see that you've made it to a trial in which treatment will be based on the RAD51 determination, on the measurement of your biomarkers to try to predict response. How are you paying for this? This is a very brave foray into a therapeutic study. This is grant supported?
This is being supported. So in cohort 1, actually, RAD51 will be analyzed retrospectively. Mm-hmm. So patients will be selected according to their mutation status. And then in cohort B, we will be selecting based upon RAD51 low score. And I mean, of course, there will be very close monitoring of tumor response in the metastatic setting. But this is actually the step forward that we want to make.
No, it's very important. We are clearly, we know that there won't be 100% response, even in the presence of the mutations that we're predicting on. So to improve our activity, our specificity is really critical.
Correct.
Judith, I have a slightly different question. I'm just interested in your thoughts. So here we've finally been able to convince pharma that it's worth having drugs for this small group of patients with BRCA1 or BRCA2 mutations. And now I saw on your slides, there's some PALB2. In breast cancer, we've sort of had these small trials adding one gene at a time. In prostate, they did a big study. They just looked at everything. How do you think we can move forward effectively and efficiently? Do we have to go one gene at a time in all of these studies? Do you think we can? Is the way to go through a predictor like RAD51, where we don't really have to know exactly which mutation is involved?
Yeah, I mean, in my opinion, what makes those tumors sensitive to PARP inhibitor is having a deficiency in the pathway. So being homologous recombination deficient. So far, tumor sequencing has been the most available and feasible analysis that we have had in clinics to identify these homologous recombination deficient tumors. But I think that now we are gaining experience and evidence that it is important that not all homologous recombination repair genes are the same. That it is not the same to have a mutation in one of the tumors that is in one of the genes that has a role in simply censoring or mediating the DNA damage and activating the pathway, that the core homologous recombination genes, such as PRCA1-2, PALB2, or RAD51C and D. And we are gaining this evidence already. So the question is, do we want to stay with tumor analysis, which is actually in clinics, we all have available, and probably in the US, you have it more than we have here in Europe. But maybe we actually want to see the consequences of those mutations. And we have seen here, it was very nice to see that was presented in ASCO 2021, that even in ovarian cancer, among this population that is so sensitive to platinum chemotherapy, among those that were BRCA mutation carriers, RAD51 could discriminate a subgroup of patients who had a better response to platinum. I mean, the question is whether this will also happen for PARP inhibitors or not. I mean, these are clinical validations to be done and to investigate, but I think the field is moving towards there. And 60% in Embraca and in the Olympiad were nice overall response rate, but they are not enough.
Right. Even in the neoadjuvants in neo-alta or neo-talo, right? 16 patients had no response at all, despite their mutation.
Correct. Correct. So we want to identify those patients.
Great. Well, Judith, that was a beautiful talk. And thank you so much for giving us your time late at night in Barcelona and giving us so much to think about. We're really excited to see your work move forward. And we hope we'll be able to become part of it in the future, or at least to see where it will take us. Thank you very much. Thank you to the Salmon family for making this talk possible. I only wish so much that we could have done it in person this year. Hopefully soon again, we'll have an opportunity for you to come and be with us so we can spend more time together. But thank you to everyone. Thank you all for your attention. Good night.
Thank you.
Good night.
Thank you. Good afternoon. Bye.